It is 11 o'clock. Ladies and gentlemen, boys and girls, this year's Nobel Prize in Chemistry deals with G-protein coupled receptors. The picture behind me shows you the latest development in a long series of developments that have taken place over the past few decades and summarizes work that has been made by this year's two laureates. The G-protein coupled receptors are an ingenious set of operators in a system that works in a modular fashion in all multicellular organisms, making it possible for one part of our body to understand what's going on in another part and to send signals to tell what to do. Rather much like the captain of an old sailing ship shouting, "Set the sails!" And you've all seen what happens in the movies. Well, some people enter the rigging, others start pulling on ropes, and half of them start singing chanties. Just from one single order. The same thing in our bodies. If adrenaline goes out into our bodies, it elicits different responses to our vision, to our muscles, to our hearts, and so on. The laureates, Robert Lefkowitz and Brian Kobilka, have been part of this development from the very beginning, and they have been leading the way. Our first lecturer, Robert Lefkowitz, was born in 1943 in New York. He received his MD from Columbia University in New York. He is a Howard Hughes investigator. He works as, you see all his titles here, the Medical Institute, James B. Duke Professor of Medicine, Professor of Biochemistry at Duke University Medical Center in Durham, New York City. North Carolina, thank you, the USA. But of course, above and all, he's a scientist. So welcome to the stage. Thank you.
Thank you very much. Needless to say, it is a real pleasure and an honor to be here with you today. The idea of receptors has fascinated scientists for more than 100 years. And it's obviously fascinated Brian Kobilka and myself for the entirety of our research careers. Now, today we know that one family of receptors that you've just heard about, the G-protein coupled receptors, also known as the 7 transmembrane receptors, represent by far the largest, most versatile, and the most ubiquitous of the several families of plasma membrane receptors. They comprise almost 1,000 genes in the human genome, and they regulate virtually all known physiological processes in humans, including our senses of taste, smell, and vision. They also are the commonest target of therapeutic drugs, accounting for perhaps 50% of all prescription drug sales in the world. Now, despite the very central position of research on these receptors in current biomedical research, it's really only in the last 30 years or so that there's been any general agreement that they even existed. Prior to that time, their very existence was a subject of much controversy and skepticism. Perhaps the earliest explicit suggestion that there might be such a thing as a receptor was made by the famous British pharmacologist J.N. Langley in 1905. He wrote, "So we may suppose that in all cells, 2 constituents at least are to be distinguished: a chief substance, which is concerned with the chief function of the cell, as contraction and secretion, and receptive substances, which are acted upon by chemical bodies and in certain cases by nervous stimuli. The receptive substance affects or is capable of affecting the metabolism of the chief substance." So here we have the first explicit suggestion of a receptor in terms of a particular item which is able to both interact with a stimulus, bind it presumably, and then act on the cell to change its actions. But this idea of a receptor was not widely accepted, and in fact was in general ignored or derided. As an example, Langley's student, ironically, Sir Henry Dale, a famous pharmacologist who actually won the Nobel Prize for his work on cholinergic transmission, had this to say 40 years later about his mentor's receptor idea. It is a mere statement of fact to say that the action of adrenaline picks out certain such effector cells and leaves others unaffected. It's a simple deduction that the affected cells have a special affinity of some kind for adrenaline, but I doubt whether the attribution to such cells of adrenaline receptors does more than restate this deduction in another form. Now, even more ironic, perhaps, is this next statement by the very famous American pharmacologist of the mid-20th century, Raymond Ahlquist. Now, Ahlquist, in 1948, published a seminal paper for which he won the Lasker Prize, in which he suggested, based on conventional pharmacological studies he had done, that there must be two types of receptors for adrenaline, which he called alpha and beta. But his idea of receptor was quite different, because 30 years later, as recently as 1973, he wrote the following: "This would be true if I was so presumptuous as to believe that alpha and beta receptors really did exist. There are those that think so, and even propose to describe their intimate structure. To me, they're an abstract concept conceived to explain observed responses of tissues produced by chemicals of various structure." He actually wrote this after serving on a symposium with me in which I presented some of my earliest work. So, it was against this background of real skepticism about the existence of receptors that those of us who were interested in bringing these mythical molecules to life began the work some 40 years ago in the early 70s. And it was clear to all of us, I think, at the time, that if we was to succeed in proving their existence, we would need to develop a whole suite of new technologies, which did not then exist. And the first of these would have to be some way of directly tagging the receptors for study. That is to say, radioligand binding experiments. And so in the early 70s, together with student Rusty Williams and postdoc Mark Carone, we set out and were successful in developing radioligand binding studies for initially the beta-adrenergic receptors and then the alpha-adrenergic receptors. And with those new techniques came the ability to study receptor regulation by a myriad of factors, the ability to discover new receptor subtypes, and interestingly, the ability to develop some interesting theories of receptor action. As an example, we found that in competition curves, antagonists, beta antagonists such as alpranolol, gave steep uniphasic curves of a single affinity, whereas agonists like epinephrine or isoproterenol gave shallow curves, which in fact were composed of two components, a high and a low affinity one. Addition of guanine nucleotides such as GTP converted all the high to low affinity receptors and now gave us a steep curve. In order to explain this initially perplexing phenomenology, André de Leon and I developed the so-called ternary complex model, which proposed that agonists combine with the free receptor to form a low-affinity agonist receptor complex. And then that complex further combines with another initially unknown component, we called it X, to form a high-affinity ternary complex, ARX. The modulatory effects of guanine nucleotides on the complex immediately suggested the idea that X might be the then very recently discovered guanine nucleotide regulatory protein. The ability of an agonist to drive the formation of this high-affinity ternary complex is in fact a measure of its efficacy or its ability to stimulate adenylate cyclase. And in fact, the ratio of affinities of the agonist for the low to the high-affinity form is a measure of its efficacy. And in fact, those affinities could be easily deduced and calculated from these competition curves. So what we had here then was perhaps the first direct demonstration of the mutual allosteric regulation of a receptor by its ligand and its effector, in this case the G protein, which led to their reciprocal ability to enhance their affinity for binding to the receptor. But of course, one of the most important consequences of developing these ligand binding approaches was that we were now able to tag the receptors to begin to try to isolate them. This was a truly daunting task. The receptors were little more than trace contaminants of plasma membranes and would require more than 100,000 purification, and then only after we could find a way to solubilize them from the plasma membranes. Our ultimate success was based on our ability to develop affinity chromatography matrices in which we covalently coupled various alpha or beta adrenergic antagonists to cephalose resins. By biospecifically adsorbing and eluting the receptors from these columns, And combining that with more conventional chromatographic techniques, after a decade of difficult work by numerous individuals in the lab, such as Mark Carone, Susanna Kotekia, John Reagan, Frederick Lieb Lundberg, Jeff Benovic, we were able to purify all 4 adrenergic subtypes, then known essentially to homogeneity. 3 of those are shown on this SDS gel. They were all, as you can see, single polypeptides, glycoproteins of molecular weight approximately 65,000 Daltons. Each of these bound radioligands with precisely the specificity that would be expected for the appropriate receptor, alpha or beta. Still, some skepticism persisted. People wanted to know, could this isolated molecule also perform the companion function of being able to activate a specific process in a cell? That is to say, to convey responsiveness to adrenaline. To address this problem, a talented postdoc in the laboratory, Rick Serione, reconstituted the receptors into phospholipid vesicles. He then took erythrocytes from Xenopus laevis, African clawed toad. These erythrocytes have adenylate cyclase and receptors, such as prostaglandin receptors. They lack beta receptors, and hence the adenylate cyclase does not respond to adrenaline. Rick fused his receptor-containing vesicles with the cells. They acquired catecholamine responsiveness. Subsequently, within a year, we were able to reconstitute the entire catecholamine-sensitive adenylate cyclase from 3 pure proteins: the beta receptor, the guanine nucleotide regulatory protein GS, and the subunit, the catalytic subunit of adenylate cyclase. This work, which was done collaboratively with Lutz Birnbaumer and the late Eva Neer, proved once and for all that our isolated molecules were truly the beta adrenergic receptor and capable of performing both functions of the receptor. Now, with validated, authentic receptor in hand, we're able to use chemical techniques, cyanogen bromide digestion. We obtained several stretches of amino acid sequence, which enabled us, in collaboration with a group at Merck, to design oligonucleotide probes, which allowed us to clone the gene and the cDNA for the beta-2 adrenergic receptor, the first ligand-binding GPCR to be cloned. This represented the first successful effort in our laboratory of a young cardiology fellow named Brian Kobilka. What we found in our paper in 1986 was that in the deduced sequence of the receptor, we could observe virtually all the now canonical features of GPCRs: the 7 transmembrane spans, the interconnecting loops, sites for end-link glycosylation at the amino terminus, and sites for regulatory phosphorylation on cytoplasmic domains. And in a remarkable surprise for us at the time, shown in blue, residues that were identical with the visual sensing protein, the light-sensing protein rhodopsin. 25 years on, people find it difficult to understand how this could possibly come as a surprise. But it really did, to all of us. The reason was that at the time, there were only 2 7-membrane spanning proteins known: rhodopsin, the sequence of which had been determined only a couple of years before, not by cloning, but by conventional protein sequencing, because it was so abundant, and a light-sensitive proton pump from primitive archaebacteria called bacteria rhodopsin. Since both of these 7-membrane spanning proteins were light-sensing proteins, it was assumed that 7 transmembrane spans must be the signature feature of light-sensitive proteins. Only with our cloning of the beta-2 adrenergic receptor did the idea begin to emerge that no, perhaps it was a signature feature of G-protein coupled receptors. Within the year, we could confirm that by cloning the alpha-2 adrenergic receptor based on John Regan's purification of the protein in the laboratory, and then the alpha-1s, and on to a total of 8 adrenergic receptors that we cloned, as well as a serotonin receptor. 3 of those based on the difficult work of protein sequencing. And so over the next few years, the idea emerged that there was a large family of GPCRs, all having 7 transmembrane spans. Over the next 5 or 6 years, the family grew rapidly, as many investigators were now able, using homology techniques based on the early sequences that we and then others obtained, to gradually enlarge the family. But almost never again was it necessary for anybody to actually purify one of these rare proteins to get the protein sequence necessary to do the cloning. And so we always felt very good about the at least in retrospect, about the very difficult decade we had spent purifying these proteins, the sequences of which actually provided a Rosetta Stone, if you will, which allowed us to clone receptors and then allow others to clone the receptors in which they were interested. We next turned our attention to using several techniques to try to understand how this highly conserved structure, 7 transmembrane spans, was specialized to perform these 2 core functions of ligand binding and activation of G proteins. We used site-directed mutagenesis and the construction of the first chimeric receptors, in this case between alpha-2 and beta-2 adrenergic receptors. Again, Brian Kobilka in the laboratory led the way in this project. Alpha-2 and beta-2 adrenergic receptors are very similar in sequence, about 50% identical, but they perform virtually opposite functions biochemically and physiologically, with the alpha-2s inhibiting adenylate cyclase through GI and the beta-2s stimulating adenylate cyclase through GS. We were able to demonstrate in these chimeras that the simple insertion into the alpha-2 receptor of little more than the 3rd cytoplasmic loop of the beta-2 adrenergic receptor converted its function from GI inhibitory to GS stimulatory. Over the next few years, much work from our laboratory and many others led to the current ideas that the membrane spans and extracellular loops were responsible for ligand binding, whereas the regions shown in blue on the cytoplasmic surface represented the regions specialized for G protein coupling. In the course of doing further mutagenesis in this area, Susanna Kotekia in the laboratory made a surprising and serendipitous discovery, which was that mutations in the distal part of the 3rd intracellular loop, which would be expected to lead to loss of function, actually in some cases led to the formation of constitutively active mutant receptors. That is to say, receptors which were active even in the absence of a ligand. At the time, we conceptualized this as being due to the fact that these mutations must abrogate certain intramolecular interactions within the receptor that confine it or constrain it to its inactive, or R form as shown here, thus mimicking the conformational changes that agonists would produce when they lead to the R star or active form of the receptor which couples to G. In just a few minutes, my colleague Brian Kobilka will show you how recent crystal structures have helped shed light on what the nature of those, at that time, unknown constraining forces and switches were within the receptor. Interestingly, we now know that a growing list of interesting diseases are caused by such occurring spontaneous and inherited constitutively active mutations in receptors. Now, contemporaneous with this work in the lab on the structure of the receptor, I had been very interested for years in trying to understand the virtually universal phenomenon of receptor desensitization, which is illustrated here for the beta-adrenergic receptor in an isolated cell system. So when one stimulates the receptors with an agonist like adrenaline or isoproterenol, cyclic AMP rises rapidly, but then even in the continuing presence of agonist, it rapidly falls almost to background levels. Here's another example for the angiotensin receptor with a different second messenger, a universal phenomenon. I've been fascinated by this phenomenon going back to the beginning of my career, and I think the reason is because it's such a classic example of what must be the most pervasive phenomenon in physiology, namely that when you perturb virtually any living system, it has myriad mechanisms which tend to return it back to its initial state. Let me tell you how we came to discover what is perhaps the major mechanism responsible for this desensitization. In about 1980, Jeff Stadel used some probes, recently developed photoaffinity probes for the beta receptor, which we had just developed, to label beta-adrenergic receptors in cells which had been desensitized by exposure to an agonist, a beta-agonist isoproterenol. And when he ran those photoaffinity probe-labeled desensitized receptors out on SDS gels, he found that their mobility in the gels was retarded compared to non-desensitized receptors. At the time, it had just been published that such retardation in gels was a fairly regular accompaniment of phosphorylation of proteins, especially membrane proteins. Accordingly, we labeled our cells with P32, and Jeff was able to show that the catecholamine-induced desensitization of the erythrocytes was in fact associated with phosphorylation of the receptor. Over the next several years, a very talented graduate student in the laboratory, Jeffrey Benovic, was able to first identify the novel kinase involved in this receptor phosphorylation. We called it initially BARK, for beta-adrenergic receptor kinase. It's now known as GRK2, or G-protein coupled receptor kinase 2. Jeff was able to purify it, isolate it, to clone its cDNA, and show that it was the founding member of a new kinase subfamily. Now, contemporaneous with this work, people studying visual signal transduction had found, in a very corresponding way, that the function of rhodopsin, when it was bleached by light, also seemed to be reduced by its multi-site phosphorylation, and that enzyme had been isolated and was being called rhodopsin kinase. We obtained some enzyme, purified it, cloned its cDNA, Wolfing-Lorenz did this, and found that it was a member of the same family. And so it became clear then that there was a new subfamily of kinases called G-protein coupled receptor kinases. Today we know that there are 7 members of the family. 2, GRKs 1 and 7, are limited to expression only in the retina. 2, 3, 5, and 6 are universally expressed. Thanks to the work of John Tesma's lab, we now have crystal structures for most of these. This is the structure done 10 years ago collaboratively with my laboratory, showing the structure of GRK2 in complex with G-beta gamma, with which it binds to translocate to the membrane from the cytosol. All the kinases share a very conserved tripartite domain organization with a highly conserved central kinase catalytic domain flanked by 2 regulatory domains. But it turned out that phosphorylation of the receptors by GRKs was not the whole story of desensitization. Something was missing. What I mean is that when Jeff Benovic was purifying this GRK2 or BARK enzyme, he found that the more he purified it, the more it lost its ability to desensitize the receptors in an isolated system. Suspecting that we might be losing something, we kept trying to isolate another molecule. We never succeeded. But then Hermann Kuhn, about that time, the late Hermann Kuhn, published a paper showing that an abundant retinal protein, which was then being called 48K protein, or S antigen, somehow worked together with rhodopsin kinase to deactivate rhodopsin. And so the protein was renamed arrestin because it arrested signaling. This sounded much like what we might be losing, although, of course, this protein was limited in its expression to the retina, so it couldn't be the exact protein we wanted. Nonetheless, I called Kuhn, obtained some of his protein, and in short order, Benovic was able to demonstrate that at high concentrations, it restored to our enzyme preparations the ability to desensitize the beta-adrenergic receptor. Then, at about that time, Shinohara at the NIH cloned the cDNA for 48K protein, or arrestin, suspecting that what we might be losing might be not just functionally analogous with visual arrestin, but structurally homologous with it. We obtained his clone, and Martin Losey, then a fellow in the lab, was able to use low-stringency screening to isolate a 70% sequence-identical protein, which we named beta-arrestin. Subsequently, Hova to tramadol, a year or 2 later, cloned another such molecule. We called it beta-arrestin 2. Now, with authentic, validated, recombinant materials in hand, and in reconstituted systems, we could show that in this upper panel, with rhodopsin kinase phosphorylated rhodopsin, arrestin was very potent in desensitizing, whereas the 2 arrestins were weak. Conversely, with BARK or GRK2 phosphorylated beta receptor, the 2 arrestins were very potent in desensitizing, and arrestin was very weak. This showed that there was conservation across the entire spectrum, all the way from rhodopsin to the beta receptor, in this desensitization mechanism. However, as you can see, there was great specificity for the nature of which arrestin was involved. Today, we know there are 4 arrestins in this family. There are crystal structures available for all of them. 2 are limited in their expression to the retina. All of them share the same fold. There are 2 domains, amino and carboxy terminal, consisting almost entirely of antiparallel beta sheets, connected by a flexible hinge region and stabilized by a polar core. So where this brought us to, by the mid-90s, was 2 paradigms shown on this simple cartoon, which are in turn based on the growing awareness that 3 families of proteins, and only 3, share the remarkable property of being able to interact in an entirely stimulus-dependent way with virtually all GPCRs. And those protein families are, of course, G proteins, GRKs, and beta-arrestins. Interaction of the agonist receptor with the G protein leads to canonical signaling through second messengers. Interaction with GRKs and berestins leads to desensitization by a steric mechanism in which the berestins block signaling by getting in the way of the G protein. But within the last 10 years, there's been a dramatic change in this paradigm, as we have begun to understand that the GRK-berestin system is actually multifunctional. Even as it desensitizes the receptors, it itself becomes a signaling system in which the berestins are able to act as scaffolds or adapters to bring numerous signaling proteins into contact with the receptors and set up signaling pathways. Some of them are shown here with their cellular consequences. Perhaps the most studied have been the ERK-MAP kinases. In addition, the berestins serve as adapters which bind to multiple elements of the clathrin-coated pit machinery, thus mediating endocytosis. So berestins, we now understand, mediate three types of functions: desensitization, endocytosis, and signaling. Now, in the course of studying this newly appreciated signaling through berestins, we made an interesting discovery 5 or 6 years ago, and that was of so-called biased agonists. Now, a biased agonist is a ligand which stabilizes a particular active conformation of a receptor, thus stimulating some responses but not others. 7 transmembrane receptor ligands, for example, can be biased toward a particular G protein or beta-arrestin. Mutated receptors can also be biased. Now, in the classic model, two-state model of receptors, an agonist combines with an inactive receptor, R, and drives it into the active R-star conformation. All signaling is then a consequence of the concentration of this unitary active state. But the existence of biased agonists, which can stimulate signaling only through G proteins or beta-arrestins, demands that there be multiple active states, shown here simplistically as 2 active states, one of which can interact with G proteins and one with berestins. By way of illustration, let me show you some simple data illustrating the ability of 3 ligands for the angiotensin receptor to promote interaction of the receptor either with G proteins, shown in red as second messenger generation, or with beta-arrestins, shown in black. As you can see, if the ligand is angiotensin, a classical, unbiased, balanced ligand, you get very nice and very similar dose-response curves for both G protein and berrestin. If you use a so-called ARB, an angiotensin receptor blocker, which are used in clinical medicine, of course they're antagonists, and so one gets absolutely no stimulation of either pathway. However, if one uses a biased ligand, this would be a berrestin-biased ligand, Trevena 027, which is an octapeptide analog of angiotensin, as shown in red, it also does not activate G proteins. That means it's an ARB. It's a blocker with respect to G proteins. But with respect to berestins, it has very potent ability to activate. Now, I mentioned to you just a couple of minutes ago that one of the major consequences of berrestin recruitment is to lead to signaling in a fashion which is parallel to, and in many cases completely independent of G proteins. So, we were interested in gaining a more global view of what signaling due to berestins might be like. And so Kevin Zhao in the lab used mass spectrometry and proteomics techniques to map essentially all phosphorylation reactions that occurred in a cell when the angiotensin receptor was activated by a purely berrestin-biased ligand, such as I showed you, which can't activate G proteins. And I don't have time to go through it, but I simply want to show you that in this very diffuse map, you can see that there's a huge amount of signaling that goes on, all these phosphorylation reactions, many of which are very similar to or identical to ones which can be activated through G proteins. This tells us several things. One, berrestin-mediated signaling is very diverse and maybe every bit as diverse as G protein signaling. And 2, many of the same pathways are activated. However, almost invariably, when the same pathway is activated, there are distinct physiological consequences to the different subcellular distributions of the activated enzymes. So in the last 2 or 3 minutes, I just want to show you why we believe this new understanding of these signaling pathways may have implications for the development of an entirely new type of therapeutic. Again, using the angiotensin receptor as a model, when the receptor is activated by its natural ligand, angiotensin, we get activation of both G proteins and berestins. But the G protein activation leads to vasoconstriction, elevation of blood pressure, which, especially in patients with heart disease, can be problematic. However, Berestin-mediated signaling can be shown to have potentially beneficial effects, cytoprotective and anti-apoptotic, for example. Now, ARBs, angiotensin receptor blockers, as I explained, are one of the most commonly used drugs in clinical medicine. They are used because they block the potentially deleterious G protein-mediated effects of angiotensin to raise blood pressure. But of course, they also block potentially beneficial effects through Berestin. So we hypothesized that a Berestin-biased ligand, which would, like ARBs, block G protein signaling, but actually preserve or even promote Berestin signaling, might have added therapeutic benefit. In fact, it's been shown that such a ligand, this 027 compound, slows progression of heart failure in animal models, lowers blood pressure, and increases cardiac performance in ways that are not true of conventional ARBs, and is also anti-apoptotic. And in fact, such a compound is in phase 2 clinical trials for the treatment of acute congestive heart failure. Now, it's also easy to conceive that a G protein-biased ligand might be what was desired in some circumstances. For example, consider opiates, the most potent pain-relieving medications available to us. It is known that their very powerful anti-nociceptive or pain-reducing effects are due to activation of the mu-opioid receptor and of the GI family of G proteins. However, there are also very nasty side effects of opiates, as you know. They lead to constipation, respiratory depression, and tolerance, which is desensitization requiring more and more drug. It turns out that virtually all of those side effects are mediated through Berestin-mediated signaling and are completely lost in Berestin-2 knockout animals. So one could conceive that a ligand for the mu-opioid receptor, which activated only G proteins and not Berestins, would have preserved therapeutic effect and markedly reduced side effects. So these are just two particular examples of how it might be possible to leverage this new understanding of these two parallel forms of signaling through G proteins and Berestins, which was gained, remember, initially through biochemical studies, how it may be possible to leverage it to therapeutic advantage. Well, I'm out of time, and of course I want to acknowledge all the many, more than 200 individuals, many of whom are here, who worked with me over the past 40 years. Just to put up a list of their names, I don't think would give a flavor of things. And so instead, I want to show you a photograph which is representative of the group. This was taken 10 years ago at a celebration of my 60th birthday party at Duke, at which many members of my laboratory from previous years, more than 100, came back to celebrate with me. And I do want to call out for special attention one fellow hidden away here in the last row. He's your next speaker, Dr. Brian Kobilka. Thank you very much.
And our next speaker here today is, of course, the second Nobel laureate in chemistry for 2012, Brian Kobilka. He was born in 1955 in Little Falls, Minnesota. He got his MD from Yale University, and he is now a professor at Stanford University. To the second.
Testing? Can you hear me? Okay. I'd like to thank the Royal Swedish Academy for this great honor. What I would like to do in the next half hour is give you a brief tour through my lab's efforts at understanding this really interesting signaling process. As Bob mentioned, the beta-2 receptor, like many G-protein coupled receptors, is a very complicated signaling protein, signaling through more than one G protein and having both signaling and regulatory interactions through arrestin. Moreover, the drugs that we work with for G protein coupled receptors often have really complex behaviors as well, ranging from inverse agonists, which inhibit basal activity all the way through full agonists. What I'm going to talk about today is really just a little piece of this fairly complex puzzle, and that is our efforts to understand the canonical signaling pathway of a G protein-coupled receptor to its preferred G protein. So the next slide I'm going to show you is something that I first saw when I came into the field, and it was pretty much at this level of detail. It's known as the G protein-coupled receptor G-protein cycle, where we start with a receptor that's inactive, an agonist binds to the receptor, promoting an interaction with a heterotrimeric G protein. This interaction then facilitates exchange of GTP for GDP. The G protein dissociates into its component parts, alpha and beta gamma, which are signaling proteins. And then the timing of this event is dependent on the hydrolysis of GTP to GDP, and the cycle begins. During the past decade, we've made some progress in understanding the different components of this. That is, what the three-dimensional structure of an inactive receptor is. We're learning something about the process of activation by agonists, and most recently, the structure of a receptor activating its G protein. And I'd like to tell you just very briefly about this really interesting story. So the outline for my talk is I'm first going to give you a brief overview of the approaches, the non-crystallographic approaches that we've used to understand structure and that have informed us a lot about how to, in fact, crystallize the receptor. I'm then going to talk a little bit about efforts to obtain crystals of the beta-2 and other GPCRs, and then finally, mechanistic insights that we've obtained from these structures. So reviewing a little bit about what Bob talked about in his lab, the first clues to receptor structure came from the clones of a number of the genes, and this is the 1986 sequence that was made possible by protein sequence obtained by Jeff Benevik. And from this nucleotide sequence, we can deduce the amino acid sequence, and then we can see within the amino acid sequence stretches of hydrophobic amino acids, which gave us clues that this was a membrane protein and a multi-spanning membrane protein that, as Bob pointed out, looks a lot like rhodopsin. And it also taught us something about the post-translational modifications, glycosylation, palmitylation, and phosphorylation. Early experiments, both chimeric receptor studies and site-directed mutagenesis, we learned that the ligand binding domain for the beta-2 receptor largely resided in the transmembrane sequences, but on the extracellular half. And domains involved in G protein coupling specificity were primarily found in the ends of the third intracellular loop. But these approaches also were used to modify the receptor to facilitate its purification. So we found that many GPCRs, and particularly the beta-2 receptor, does not have a natural cleavable signal sequence. And by inserting a signal sequence and subsequently an epitope for an antibody, we could both enhance the expression and overall processing of the protein and facilitate its purification through the use of an antibody column and at the end of the protein through a nickel affinity column. And using these approaches, we were able to use a multi-step purification process, initially expressing receptors in insect cells, solubilizing in detergents, using an antibody affinity column, subsequently a ligand affinity column. Much of the protein that's made in insect cells is not functional, so this is a key step in separating functional receptor from non-functional receptor. And then finally a cleanup step, a nickel column, and we end up with pure protein that's almost 100% functional. And this can be used for a number of different types of studies, including biochemistry, spectroscopy, and crystallography. Very briefly, what we learned from biochemical studies is shown here, mainly through the use of proteases. we identified fairly large stretches of the protein, the amino-terminus, the third intracellular loop, and most of the C-terminus that were susceptible to proteases, suggesting that they were unstructured or flexible. And this is something that's useful to know when you're trying to crystallize a protein. But I think one of the areas that we learned most about the protein was in the application of spectroscopy, particularly fluorescence spectroscopy, and more recently EPR and NMR spectroscopy, to help us understand dynamic properties of the protein. And we learned, as I'll show in a couple of examples, that there are ligand-specific conformational states, that the receptor is very dynamic and flexible in character, and that these spectroscopy tools that we used ended up being very useful in monitoring receptor activity, particularly in crystallography procedures. Our favorite model system involves labeling cysteine 265 at the end of transmembrane 6 with any of a number of fluorophores. And I'm going to show you two early examples and the types of experiments that we did and the types of conclusions we reached. So this is the simplest experiment, type of experiment, and early on these types of experiments were first performed by Ulrich Gether, but this one was performed later in 2004, and we simply are labeling the beta receptor, a purified beta receptor with a fluorescent probe, in this case rhodamine, and that rhodamine is tucked in the environment of the receptor, and when the structure of the receptor changes, the environment around the fluorophore changes and its intensity changes. So you can see here, time 0, we add agonist, and we have this really nice change in the intensity of the beta receptor as a function of time. But this wasn't a simple exponential function. It actually was best described as a biphasic process with a rapid phase, and this is somewhat limited by the experiment, the mixing time. So there's a rapid phase and a slow phase. And as Bob showed, functional assays early on where we tried to model the receptor with a simple two-state system, an inactive and inactive system state. And as Bob pointed out, functional studies were beginning to suggest that this was too simple. And these experiments, in fact, were more compatible with a sequential model of conformational changes, agonist bind, some possible intermediate state, and then a final one or more active conformations. So this is one of the first biophysical evidence we had for multiple ligand-specific states. Similar experiments were done using the same model system, cysteine 265, now labeled with fluorescein, and a different spectroscopic technique, which is fluorescent lifetime analysis. And the lifetime of a fluorophore, as its intensity, is very sensitive to its environment. But if one has different conformations, one can often see multiple lifetimes at the same time. So, for example, this is a distribution of lifetimes in receptor in the absence of any drugs. We call this basal. And you can see it has a single peak that's fairly broad, and this would be compatible with predominantly one conformation, but a very flexible conformation. When we add the neutral antagonist, which binds to and competes for other ligands but doesn't change basal activity, it doesn't really change the fluorescent lifetime, but you can see it narrows the distribution, suggesting that its receptor might be becoming less flexible. What's particularly interesting, if we now add an agonist, isoproterenol, we see two distinct lifetimes. Again, compatible with what we saw on the previous slide, two agonist-specific conformational states. And as I'll point out again, it suggests that an agonist alone can't fully stabilize an active conformation. Now, if we take another kind of agonist, a partial agonist that has some different functional properties, and look at its behavior, we see, like isoproterenol, it has two peaks. And the peak for which we think is the active conformation is different from the peak for isoproterenol, suggesting that, again, yet another conformational state, that the active state for this partial agonist is different from the active state for the full agonist isoproterenol. So we have evidence for multiple agonist states and ligand-specific conformational states. So these these experiments are part of a large number of experiments that we've conducted over the years, more recently including NMR and EPR spectroscopy experiments, from which we have the following conclusions that are both interesting of themselves and also relevant for crystallography. And the first is that the beta receptor is flexible and dynamic. And as an example of that is this is a molecular dynamic simulation provided by Ansgar Philipsen and Ron Dror at the Shaw. And this really gives you a sense of the dynamic character of the protein. This is about a 30 microsecond simulation. We found that transmembrane 6 undergoes the largest changes in response to agonist. Agonist binding and activation occurs through a series of conformational intermediates, as we've seen before. Agonists and partial agonists stabilize distinct conformational states. What will come up again is that agonists alone do not stabilize a single active conformation, and this is important again for crystallography. And then finally, fluorescence spectroscopy particularly has aided in identifying the optimal conditions for crystallography. So now I'm going to move forward and begin to discuss our efforts to crystallize the beta receptor. And I would say our serious efforts began at the end of 1990s and early 2000s when we were able to make sufficient quantities of protein to start crystal trials. And our initial efforts involved trying to crystallize the inactive conformation bound to an inverse agonist. And this was one of the first really exciting observations that we had. These were crystals that we generated in around 2004 with what we consider the wild-type receptor. It had these tags on, but otherwise it was pretty much unmodified. And we were pretty excited. We were helped in this effort by Gebhard Schertler, who gave us time at the microfocus beamline in ESRF because these crystals were so small and weakly diffracting that we otherwise wouldn't have been able to tell whether they're a protein or not. But we got this very, very weak diffraction pattern, which was compatible with protein, but only 20 angstroms. Nevertheless, we were extremely excited at this point because we had our first protein crystals, and we thought that it's just a matter of weeks, but it wasn't. So what was the problem? Well, let's take a hypothetical GPCR that has uniform conformation, really nice structure. If you have purified protein and the structure is uniform, it should be possible to get high-quality crystals because all of these proteins will stack together in the same way. What we had, though, was a conformationally heterogeneous protein, and our spectroscopy studies showed us that at least transmembrane 6 was quite dynamic and also heterogeneous. So now we have purified protein. It looks nice on a gel, but these molecules don't really fit together very well, and the crystal quality is poor. So in addition to the challenges of protein dynamics, we also had the challenge of the limited amount of polar surface area. Most of the protein was embedded in a membrane or for purified protein in a detergent micelle. And so around 2005, 2006, we started working on efforts to overcome this limitation. And we took two approaches. And I was very fortunate to have two very talented postdocs join the project at the time, Søren Rasmussen and Dan Rosenbaum. And they took two complementary approaches and actually worked together on both of them. The idea was to stabilize the dynamic component of the protein and also provide additional polar surface. Søren chose to identify conformationally selective antibodies that recognize this region of the protein in its folded form. And Dan chose protein engineering. And very, very quickly, they both succeeded very close to each other in time. And these two structures were published in 2007. The beta-2 Fab structure and the T4L, T4 lysozyme fusion structure, it was really reassuring when they both came out together because we used two different approaches and the structures were very similar. We lacked some of the definition in the extracellular domains, but the intracellular components were virtually identical. In addition to these tricks that we use to help get the structures, another important component was using lipid-based media. The original crystals that I showed early on were also obtained in a mixture of detergent and lipid. The receptor fab crystals were grown in biocells, and these crystals were grown in lipidic cubic phase, and this was done in collaboration with Vadim Cherzov and Ray Stevens. More recently, in an effort to get our receptor G protein complex, we found that you can even put T4 lysozyme on the outside of the receptor and not have, and simply truncate the third loop, and you can get crystals in this way as well. So this is a summary of the crystal structures, the inactive state crystal structures that we've gotten recently. And the T4 lysozyme fusion has been the most successful in our hands and probably in the field, at least so far. You can see the muscarinic receptors, two muscarinics, two opiates, and the protease activated receptor all were obtained using this method. Also in the Stevens lab at Scripps, a number of other GPCRs have been solved using this fusion protein strategy. However, I want to point out that other methods have worked, and this includes the use of thermostabilizing mutations to tame some of the dynamic character of the protein. Has succeeded in obtaining crystal structures of the beta-1 adrenergic receptor and adenosine A2A receptor. This is work from Chris Tate and Gebhard Schurtler and their colleagues. And I simply must point out that rhodopsin was first. And rhodopsin so far is the only GPCR for which we've been able to obtain crystals from native protein. And in fact, the very first structural information we have comes from Gebhard Schurtler's 2D crystals, where he obtained these 2D crystals in native rod outer segment membranes. And the crystals that were obtained, the 3D crystals from Palczewski and Okada, also were grown in a mixture of lipids from the retina and detergents. And more recently, the active-like structures from Ernst and Hoffman were also grown out of protein purified from rod outer segment. So these, we believe that rhodopsin has an unusually stable character that allows this type of crystallography to be performed. So I now want to get back to our efforts to obtain an active structure. And the first question that we had is, what do agonists do? Can we actually trap this state, an agonist-bound state, on its way to activating a G protein? And this we worked on for quite a while with little success. We were not able to obtain any crystals of a receptor bound only to even a very high affinity agonist. And the first structure that we ultimately were able to obtain was one generated by Dan Rosenbaum with the help of Ralph Hull and Peter DeMiner, who developed a covalent agonist for the beta-2 receptor. And this crystal structure was obtained, but it was in an inactive state. So this gets back to what we were learning from EPR, NMR, and fluorescence spectroscopy, and that is agonists alone don't fully stabilize an active state. So this is a cartoon of a GPCR. I only have 2 transmembranes to illustrate it. But what I show here is that the receptor is flexible to begin with, and when you bind an agonist, it appears that the dynamic character actually increases rather than stabilizing a uniform conformation. And so to stabilize a uniform conformation, you need a stabilizing protein interaction. If this isn't a G protein, then you need to find some other surrogate. And a couple of years ago, I had the good fortune of meeting Jan Sterrt, who's, I think, here in the audience, who introduced me to nanobodies. And I can't go into these in detail, but they are remarkable. So they're the variable domain of single-chain camelid antibodies. And by camelids, that include llamas. And this is Pipette, the first llama that helped us to get a crystal structure. And so as a stabilizing protein, we were able to get the first active state of the beta receptor. And I'll just quickly say right now that this nanobody stabilized state is extremely similar to the G protein state that I'm going to talk about in a minute. Something we haven't published yet is a partial agonist bound state, again, stabilized by a nanobody that selectively prefers receptor bound to a partial agonist. So now on to the challenge the true active state, the receptor G protein complex. And this is a project that's been going on, I believe, since around 2006, when Roger Sunahara and I began working together on trying to understand how we can investigate the interactions between these two proteins and hopefully try to get a structure. And this has been a really fantastic collaboration that continues to date. This turned out to be a lot of work and very challenging. And I really don't have time to go into this in detail, but I think it's useful to illustrate or to at least discuss some of the important contributions to ultimately getting a crystal structure. So the first involved being able to identify a very high affinity agonist. And most of the agonists have, even the best agonists have off rates measured in seconds or minutes. Søren Rasmussen screened compounds, over 60 that were provided by various pharmaceutical companies, and found one that had a dissociation half-life of around 30 hours. And this has been, I think, a really important component of the success. The other important component is getting rid of GDP. The complex that we form that's extremely stable is nucleotide-free. And even a small amount of GDP that dissociates, or certainly any other GTP, destabilizes the complex. And Roger and his group came up with the clever idea of adding a pyrase to degrade GDP, and it really is fantastic at stabilizing the complex. Other important contributions are the development of new detergents in collaboration with Pilsup Che and Sam Gelman. We had access to these detergents, and they've really helped us stabilize the complex. We had access to unconventional lipids for lipidocubic phase from Martin Caffrey. These were critical. We also, again, had the help of Jan Steert, nanobodies that helped to stabilize the complex, protein engineering, T4 lysozyme on the outside, and finally, a really critical component to the project was the use of negative single particle EM studies to help guide our crystallography efforts. And to really cut to the chase, we finally succeeded in early 2011. And these were the first crystals grown in the specialized lipidocubic phase that were roughly, these are relatively large crystals, in fact, but we were fortunate to have access to the Argonne National Lab microcrystallography system. And this is the crew, after coming back from our last synchrotron trip, obtaining our final data set. And this is Andy Cruz, Brian DeVries from Roger's lab, and Søren Rasmussen. So I'm going to, in the last 5 minutes or so, summarize what we learned about the structure. So this is the cartoon, and now we have a three-dimensional picture, and there were some surprises. So first of all, what I'd like to do in the final few slides is take you from agonist binding to conformational changes on the inside of the cell to conformational changes in the G protein. And in this series of slides I'm going to show, the active structure is green, the inactive structure is gray. Remarkably, there are a few very, very subtle changes in the extracellular surface with larger changes in the cytoplasmic domain. So binding here amplifies conformational rearrangements on the intracellular side. The largest change is seen at the end of transmembrane 6 as evidence predicted from fluorescence experiments. And you can see it's about 14 angstrom outward movement. This is from the inside of the cell. If we start at the binding pocket, this is the inactive state bound to the inverse agonist caracolol, and the active state bound to this special high affinity agonist. And when I'm toggling back and forth between these two, if you look at any one amino acid, it's really, there are very, very subtle changes. But what you can see is the overall binding pocket becomes a little smaller, and I don't really have time to show it, but the extracellular surface of the binding pocket closes over. And ligand dissociation, even even small ligands such as isoproterenol and epinephrine, their dissociation is drastically reduced because of these changes. And this can explain the high-affinity state that Bob mentioned. The G protein-dependent high-affinity state for agonist has a lot to do with the conformational changes that occur on the outside of the receptor that are transmitted up from the G protein. So the changes in the binding pocket are really quite subtle, amounting to about 2 angstrom movement, the largest movement around serines 207, which had been identified in mutagenesis studies as being important for agonist activation. So we wanted to try to see where these changes propagated into larger changes. And right below the binding pocket, there are packing interactions with highly conserved amino acids in the family A GPCRs, including a proline in 211, an isoleucine in transmembrane 3, so this proline is in transmembrane 5, highly conserved, phenylalanine 282 in transmembrane 6, highly conserved, and asparagine in transmembrane 7. And I've gotten rid of the other transmembranes just for clarity. These packing interactions are really important for maintaining the receptor in the inactive state, and a simple movement of 2 angstroms at the top near the binding pocket is incompatible with this packing interaction, and a new arrangement is stabilized as a result of agonist binding. And so you can see, toggling back and forth, a small change in the binding pocket, a fairly large change on the intracellular side, sufficient to accept a G protein or an antibody. As I mentioned, though, the coupling between these is not rigid, it's relatively weak, and we've not been able to get a structure of this really high affinity agonist bound to the receptor alone. We need something here, and in this case, the stabilizing protein is not an antibody, but is the G protein GS. So this, in the last few slides, I'm going to show you the structural changes that occur in the G protein as it engages the receptor. So this is the blue, in this case, blue is inactive model, a model of inactive GS. There are 2 major domains, the RAS-like domain and the alpha helical domain. And between these 2 domains is the nucleotide binding pocket. And there's some really interesting details about structural changes that occur here that I don't have time to talk about, but you'll see major structural changes that do occur that are fairly easy to understand. So first of all, I'll now toggle to the active state, and I'm going to toggle back and forth. And you can see the largest structural change is a very large movement of the alpha helical domain and opening up of the nucleotide binding pocket, which the nucleotide is then released. The other very large change is the C-terminal alpha helix, which projects into the core of the receptor and stabilizes it in this active state. So this is the guanine nucleotide-free, very high-affinity interaction. We were somewhat concerned about this very unexpected large movement of the alpha helical domain. And fortunately, as I mentioned, and this just summarizes this movement, about 130-degree rotation of this alpha helical domain. And this was really quite unexpected. Fortunately, as I mentioned, we had been collaborating with Yorgos Skidiotis at the University of Michigan, and his graduate student obtained a series of really beautiful single-particle negative state images, which, and fortunately, we had tags that allowed us to identify the components of this density as being receptor, a beta subunit, and an alpha subunit. And what he was able to show is that, in fact, the alpha helical domain is very flexible and assumes many different conformations in a non-crystallographic preparation. So, in conclusion, we've learned a lot in the past couple of years about this really important interaction, the activation of a receptor in a G protein. But there's a lot we still don't know. One is how these two proteins prefer each other, why a receptor, such as the beta receptor, prefers GS over GI. We also know little about the structural details of how a receptor is phosphorylated by GRK or interacts with arrestin, and there's a lot of work to do in the future. So I'm going to conclude with showing you this is a picture taken last December at the GPCR workshop in Maui. This is Many members of the Beta 2 GS team, and many of whom are here. Here's Roger Sunohara, I want to point out, Tungsan, Bill Weiss, Yorgos Skiniotis is here, Jan Stair. Of course, I believe Soren is here as well. And my lab manager, Fun-san. And I just want to put up at the end, there are way too many people to really to have time to thank. But I want to especially thank my wife, Tungsan, who has been my colleague, friend, collaborator, a great mother. And if you've read anything about me, you know that she's key to any success I've had. I want to thank Bob and the colleagues in the Lefkowitz lab. I want to thank particularly the people who really helped me out when I started in the lab as a very green scientist. Ruth Strasser and Susanna Katekia really helped me learn how to do some things like binding. Jeff Benovic and John Reagan and Mark Carone really taught me the ropes of protein chemistry. Finally, my lab students, postdoctoral and fellows throughout my time at Stanford, and my two closest colleagues, Bill Weiss at Stanford, who is starting to teach me how to understand the process of solving crystal structures, but I'm still working on it. And Roger Sunohara, a very good friend and colleague who's been working on G protein signaling with me for a number of years. And this is the Beta 2 GS team. I want to thank my financial support from the NIH, generous gifts from the Mathers Foundation and Lundbeck. And I just want to say a colleague of both Bob and I, Virgil Woods, who's a really fantastic mass spectroscopist, passed away this year, and we'll miss him greatly. So I want to thank you for your attention.
So let us take this opportunity not only to thank the two lecturers, but also to congratulate the two laureates.